Inside every living cell lies a secret code, DNA, the blueprint of life. But how do scientists prepare this invisible molecule from the crowded world inside a cell? This process is called DNA isolation. It is a fascinating journey from breaking opening the cell to collecting pure strands of genetic material. So there are various steps. The first step involves the breaking open the cell. So every cell protects its DNA inside by its membrane made out of lipid and proteins. To release the DNA, scientists first break these barriers using detergents or lysis buffers, and the detergent function is to dissolve the cell membrane and nuclear membrane, freeing the DNA into solution. At this stage, the mixture contains not only DNA but also proteins, RNA, lipids, and cell debris. And think of it like opening a tightly packed treasure chest hidden inside the cell. Next step is the removing protein and impurities that is there. Now comes purification. Phenol or chloroform extraction is used to separate proteins from DNA. When phenol is mixed with the cell extract and centrifuged, the solution forms different layers. The upper aqueous layer contains DNA and RNA. The middle interface traps coagulated proteins, and the lower phenol layer contains unwanted impurities. To remove RNA, enzymes called RNase may be added to digest proteins, and proteinase K is often used. Slowly, the DNA becomes cleaner and purer. Basically, when I carried out my experiments, I used RNase last step after finally isolating the DNA, and proteinase was the enzyme that was used in my experiments. So in case of next step, that is there is DNA precipitation with ethanol, and DNA is soluble in water but not in alcohol. So scientists add a cold ethanol or isopropanol to precipitate the DNA. As alcohol is layered carefully over the DNA solution, white thread-like fibers begin to appear. And while these strands are actual DNA molecules clustering together in concentrated sample, the DNA can be even wound around a glass rod like a soft cotton fibers. For dilute samples, centrifugation helps collect DNA as a pellet at the bottom of the tube. So, understanding the DNA pellet after centrifugation, scientists observe the DNA pellet carefully. A clean white pellet usually indicates good quality DNA. But the thing is, like, but sometimes bubbles appear in the pellet. This may happen due to air trapping, vigorous mixing, residual detergent, low DNA concentration. In such cases, the sample may be washed again with 70% ethanol and centrifuged carefully. A smooth, clear pellet is the sign of successful DNA isolation. And DNA isolation matters because DNA isolation is the foundation of modern biotechnology and modern molecular biology. It helps scientists in genetic engineering, PCR and sequencing, disease diagnosis, forensic science, evolutionary studies, and even in agriculture as well as crop improvement. Without DNA isolation, many discoveries in genetics and medicine would not be possible. So, from a hidden molecule inside the microscopic cells to visible white strands floating in alcohol, DNA isolation reveals the huge language of life. Every pellet collected in laboratory tube holds the story of an organism in the past, its present, and its biological future. And to isolate DNA is to uncover the instruction manual written inside every living cell. Thank you.